In our body, every cell contains genome that is made up of approximately 3 billion nucleotides. And in these nucleotides, there are more than 30,000 genes are present. DNA is a double-stranded molecule. These strands are attached with each other by hydrogen bonding. There are 4 bases: adenine base pair with thymine and guanine base pair with cytosine. Due to advancement in science, the researchers identified such thousands of genes which causes risk in the disease. To understand this, that how genes work, the researchers needs to control the methods of genes. In living cell, it is not easy to change the genes, but recently a new method is discovered by which we can easily edit the genome of any species. This method is called as CRISPR method. CRISPR method is basically taken from bacteria, which is natural, by which bacteria protect itself from viruses. When bacteria detects any virus in its body, it produces a RNA molecule in which there are sequences that are matched with viral DNA. This RNA is called as guide RNA. This RNA make a complex with protein that is called as Cas protein. Cas protein is a nuclease protein that works as an enzyme and can also cut DNA. When matching sequence of guide RNA pairs with target sequence on viral DNA, then Cas protein active and cut the viral DNA, by which viral DNA become non-functional. By this method, bacteria protects itself from viruses. After studying this method, scientists realized that CRISPR system not only cuts viral DNA, rather by using this method in any living organism, we can cut target location of our interest. And when the DNA of cell cuts, then cell tries to repair that cut, but mostly in repair process, sometimes mutations occur due to errors, due to which if the cut region is off gene, then gene becomes non-functional and turns off. And thus we can turn off some genes through CRISPR method. Beside that, by using the CRISPR method, scientists after cutting the DNA can insert designed DNA of their own interest. And the most special thing is that it can not only performed in test tube but also in direct living system. By using this technique, if we do gene editing in stem cell, then genes are transferred inside all the cells made from stem cells and perform its functions. Beside this, CRISPR method can also be used in fertilized egg, and by this we can insert or change genes of our wish in embryo and baby. And CRISPR is such a powerful tool that many genes can be targeted simultaneously. And the biggest advantage of this method is that we can control diseases which can cause by multiple mutations like cancer. Along with this, CRISPR method is not empty from ethical issues. Because of this technique, designer babies can be born which have the abilities of our will. As they are very intelligent, very beautiful, very strong, less diseases can cause, etc. Some people say that it is not a bad thing to insert these qualities in next generation. Because of this, the world will develop more because of these talented children. But of course, these qualities will challenge the nature.